Nucleic acids are central to much of modern biochemistry and the medical field. We will rapidly go through talking about the structure of nucleic acids, beginning with the simple bases and advancing to the more complicated structures. Nucleic acids are linear polymers of nucleotides which fall into two categories, DNA and RNA. DNA functions as an information storage molecule, and RNA is involved in information transmission. Nucleotides are defined as a nitrogenous base plus a sugar plus a phosphate. There are two classes of bases called pyrimidines and purines, and this slide shows the numbering scheme of both pyrimidine and purine, which is useful in talking about the structure of the nucleosides and nucleotides. The most familiar purine is that of adenine, which is also known as 6-aminopurine. Guanine, or 2-amino-6-oxypurine, is the other purine. There are three pyrimidines. Uracil is used only in RNA, thymine is used only in DNA, and cytosine is used in both. Note that uracil and thymine are very similar in structure, with the thymine having an extra methyl group. There are a few physical properties of bases that are important to touch on. First, they are planar as a result of sp2 hybridization. Second, because they are conjugated, they absorb light, 260 nanometer UV light. This property is used to quantitate nucleic acids. Third, they are water-insoluble compounds at physiological pH. Fourth, the bases themselves are acid and base stable. Lastly, they exhibit a kind of tautomerism, which was important as Watson and Crick were developing their model of DNA structure. Nucleosides are bases that have a sugar covalently bonded to them. Make sure you understand which sugar it is that is present in each type of nucleic acid. Nucleosides can exhibit anti- and syn conformations, of which the anti-syn conformation is widely used in textbooks to save space, but the anti-conformation is the physiologically relevant form. Notice that also that we are numbering the sugar ring, but to differentiate it from the base numbers, we use a prime to indicate that we are talking about the sugar. It is important to note that either ribose or deoxyribose can be attached to the base through an N-glycosidic linkage, and that the name of adenosine adenine becomes adenosine when the sugar is attached to it. Nucleotides have one or more phosphate groups attached onto the base plus sugar nucleoside. The name of the molecule has to change to indicate this altered structure, and adenosine becomes adenylic acid. On this slide, I am showing the structure in the protonated form, but the pK value is about 1, and the structure is negatively charged at physiological pH. Nucleotides are joined together by phosphodiester linkage to construct the nucleic acid. On this slide, I am showing a somewhat incorrect simplification of phosphodiester bond formation. I am also using yellow plus blue makes green to show why we call this a phosphodiester. Nucleic acids have a primary structure, which is defined as the sequence of nucleotides, as well as a secondary structure or three-dimensional shape, which is the alpha helix in the case of DNA, as well as a tertiary or global three-dimensional shape. Base pairing occurs as A and T form 2 hydrogen bonds, and G and C form 3 hydrogen bonds, as shown here. You should be able to correctly indicate the functional groups that are involved in these hydrogen bonds. The most common type of DNA secondary structure is the bDNA form of an alpha helix, shown in figure 4.12 of your book. The AT and GC base pairs form the center of the alpha helix, as shown by the blue spheres in this figure. In red and purple is the phosphodiester backbone. bDNA prominently displays a major groove and a minor groove that are indicated in the figure. Both of these are important. For example, proteins can read the base sequence by sliding along the major groove. It is also important to point out that there are 10 base pairs stacked in each turn of the right-handed alpha helix. The diameter of bDNA is 20 angstroms, or 2 nanometers in diameter. These dimensions are important in order to emphasize that DNA is a very large molecule. Figure 4.18 from your book shows a bacterial cell which has been lysed in order to release the DNA. This emphasizes that DNA must be packed in order to fit into the cell. Prokaryotes use a method of packing referred to as supercoiling that I will not talk about further. Eukaryotes and, as it was published in 2017, archaea use a different method of packaging, which is to wrap DNA around proteins. Eukaryotes use a protein called histone, of which there are 5 different molecules. Figure 21.11 of your text uses a simplified diagram which does not readily show the fact that a nucleosome is composed of 2 copies each of H2A, H2B, H3, and H4. This histone octamer has 1.75 turns of DNA wrapped around it. By electron microscopy, these nucleosomes look like beads on a string. In the presence of histone H1, the nucleosomes condense onto each other to form a structure referred to either as a solenoid or 31 nanometer fiber. This is represented as the blue rod-like structure on the lower left-hand side of the figure. Other protein scaffolds cause these solenoid fibers to loop around it. This is similar to what happens if you tighten or wind up a rubber band in an extended form and then allow the ends to approach each other. At any rate, the result of this is that the chromosome looks fuzzy in an electron micrograph image, and I would encourage you to use Google or whatever your favorite search engine is to look at a structure of a chromosome. The nucleosome, solenoid, and other higher order structures represent the tertiary structure. It should be pointed out that although the B form of DNA predominates, there are other forms of DNA which have been observed. zDNA is perhaps the most important of these, as it exists in AT-rich regions of DNA, which are more transcriptionally active, and this is thought to be involved in regulation of protein expression, as we will see at the end of the course. aDNA is observed when the humidity decreases to 75%. There is some question as to whether this is simply an experimental artifact or whether it is physiologically important. The same can be said for other forms which develop by changing salt type, salt concentration, and/or humidity. Another interesting but perhaps irrelevant form of DNA is a cruciform structure which forms only in palindromic sequences. Two other short topics covered in your book that I will not talk about are Hooke-Steen base pairing, which results in triple helices, and G quadruplexes. It is briefly worth noting that DNA is commonly referred to as melting. The term melting is technically not correct, as a solid is not being converted into a liquid. However, heat does cause a change in the absorbance observed for DNA at 260 nanometers. This is explained on the molecular level by the breaking of the hydrogen bonds holding the two strands of DNA together. One of the most important applications of melting is in the experimental process known as polymerase chain reaction, PCR. Please read the preview of the genetic biochemistry section, which is straightforward.